what you did set up today. You set up one of them today. So now you're going to understand what you set up today. Recombination or recombinant. When you make a recombinant, what that means is you created a new life. That's what it means. Bacteria that acquires DNA, or any organism that acquires DNA from another organism, is called a recombinant. That's exactly what you did today. I gave you E. coli on the plate, you added plasmid DNA in there. Walking out of this room, be proud that you created new life. You created a recombinant. It didn't exist until you did what you did. And we'll see if you succeeded at it next week. If it fluoresces, you succeeded. So how are recombinants produced? Well, one way is called horizontal gene transfer. We talked about one of them before, conjugation. When we talked about the pilus, we said two bacterial cells conjugate. The closest to bacterial sex is conjugation. The pilus is formed. Excuse me. The channel between them is formed. And the plasmid, a copy of the plasmid, gets transferred over. We'll see that in the slide form. But there are other two things, transformation and transduction. This is what you did today in the lab. When you added the plasmid DNA into E. coli, you did what is called transformation. Let's look at it one at a time. All three of them together is called horizontal gene transfer. So we did this before. A donor cell copies its plasmid, sends it through the pilus to our recipient cell. We've done this before. We've talked about it before. Notice what the donor cell does. The donor cell is not going to give the plasmid to the recipient. No, that's my plasmid. I'll give you a copy, but you're not taking my plasmid. So it copies it, passes it through the pilus, and now the recipient cell is now a recombinant. It's a new life. Before and after. Before, it did not have the plasmid. After, it did. So now this one is called a recombinant. It now has new characteristics, new phenotypes, new traits acquired through the plasmid. One of them could be antibiotic resistance, for example. This may not be antibiotic resistant, but this probably is. All right, here's another type of conjugation. Some of you will swear to me that I never talked about this while I'm talking about it. What does that mean to you? It's on the exam. There you go. High frequency recombination or transfer. HFR transfer. It's a fancy type of conjugation. Notice here that the only thing transferring here is the plasmid. That's it. A copy of the plasmid. And notice also that the plasmid never becomes part of the main chromosome. There's a name for that, and it's called extra chromosomal. Extra means outside chromosomal of the main chromosome. So when the plasmid is never part of the chromosome, we call that plasmid extra chromosomal. But sometimes the plasmid is incorporated into the main chromosome. That's like this one. When this one gets copied, notice something. It copies a piece of the donor chromosome along with it. It copies the plasmid, but also a piece of the donor chromosome as well. That's the high frequency transfer, HFR. Follow through. Look what the new recombinant has. The new recombinant has its original chromosome, the purple, has the plasmid, and now has a piece of the donor chromosome as well, bacterial. So obviously this one will probably get more traits than this one would, because it's got the plasmid plus a piece of bacterial chromosome. But it too is a recombinant. All right, the one you did today is this one, transformation. You added transformation reagent today, am I right? 250 microliters of that. Then you followed it up by adding the DNA, you added the bacterial cells, and then you heat shocked. Do you agree with me?
Yes.
Good. The combination of those things does this. Your bacteria in your tube would look like this. The plasmid that I added would be outside. It's called exogenous DNA. DNA that's outside of the cell. So the E. coli that you took off of your plate would be like this one. The DNA that I gave you is like this one. It's exogenous DNA. It's DNA outside of the cell. Now, this bacteria will not take up that DNA until you make it competent. When you add the transformation reagent to the bacteria and you heat shock it, you have to do both of them. You make the bacteria competent. What does that mean? Meaning willing or able or capable of taking in exogenous DNA. That's what it means. If you give me $100 and I say I can't take it, then I'm not confident. But if I do take it, then I'm confident. The acquisition of DNA from the outside makes the bacteria competent. Yes?
Why does it break a protein?
A very good question. Here's what happens. When you add the transformation reagent, you're making the cell membrane weak, slightly weak. When you heat shock it, the cell membrane all of a sudden goes like this, like that. It opens up a pore. You know, think about you sitting on ice and then falling into the jacuzzi. Oh, you'll be like that. Well, it's the same thing. They get shocked from that transfer from cold to hot within seconds. And their cell membrane reacts to that and it opens a pore and the DNA sneaks in. It's a pretty clever technique. But it has to be the combination of transformation reagent, which is calcium chloride. It's a salt that makes the cell membrane weak. But then heat shock will cause the pore to open up and the DNA sneaks in. Now, not all the bacteria is going to get the DNA, but hopefully at least 40-50% of them will, and then you'll get some success next week. So you have to make the bacteria competent or capable of taking in exogenous DNA, and we do that in the lab by adding the transformation reagent followed by heat shock. Beautiful question on the lab exam. Now in nature, this can happen in your gut, for example. What happens is this. Bacteria, for example, dies and lyses. It lived its life, and it died, and it lysed. Its chromosome is broken into pieces. Now it's in the nearby environment of this bacteria. This one is still alive. It takes in a piece of that DNA, whatever gene is on there is now going to be part of this bacteria. Now this is now a recombinant. And it happens in your gut all the time between the bacteria that live in your gut. All right, there's another kind called transduction. Transduction involves a bacteriophage. Every time you hear the word transduction, we're involving a bacteriophage here. The virus that infects only what? Bacteria. Here they are, the alien-looking guys there. They will still inject the DNA. This is, now watch the color scheme here, please. I think this is maroon, am I right? The maroon colored DNA is viral. The pink one here is bacterial. So this is the donor cell's bacterial DNA. This is viral DNA. The virus will do what the virus does. It will absorb, it will penetrate by injecting the DNA into the bacteria. And like we said before, the virus says, the bacteria is now mine. I'm going to order you to make more viruses. Remember all that, chapter 6? Right? Now, it decides I need AGCT, the building blocks of DNA. And it realizes there's a whole bunch of AGCT in that bacterial chromosome. So it's going to order the bacterial chromosome to break apart so they can use the monomers to make the viral DNA, the maroon one. But notice something. Look at the picture. Look at the slide. Not all the bacterial DNA breaks completely. Just by chance, a piece of the bacterial DNA says, no, I'm not going to break down any further. That's it. That's it. You can break everything else down, but not me. This is a chance event. So again, the virus is going to ignore that, and it's going to start packaging. I have a capsid, I'm going to put viral DNA in there. I have a capsid, I'm going to put viral DNA in there. I have another one, I'm going to put viral DNA in there. And I have this one, I'm going to put DNA in there. I don't know, is it viral? I don't care, just put DNA in there. See, the virus, when it goes to assemble, it has to put DNA in the capsid. It can't tell the difference between viral DNA and the bacterial DNA. So by chance, you have 4 viruses. 3 of them have viral DNA in there. 1 of them has bacterial DNA. Watch this. It will lyse like it normally does. This virus with the maroon DNA will infect another bacteria and force it or order it to make viruses. This one will do the same thing, this one will do the same thing because they have viral DNA in there. It's this one that we have to talk about. The one with the bacterial DNA, but remember, by chance, you put a piece of the bacterial DNA in this virus. And it will go and infect another bacteria, but this time it's injecting bacterial DNA, not viral DNA. All this bacterial DNA is going to do is integrate into the chromosome to create a new recombinant. It's not going to create new viruses because it doesn't have that. It doesn't have the viral information. It has bacterial genes on there. So now you have a new recombinant with additional bacterial genes. Which genes? I don't know. Maybe antibiotic resistance or something. I don't know. It's a random thing. So all 3 of them, if we go back, are collectively called horizontal gene transfer. And that's how bacteria produces new recombinants, whether in nature or in the lab. Like transformation, we have control over to do that in the lab. Like you did. Now, your bacteria, the result from your transformation experiment is going to produce a recombinant that fluoresces green under ultraviolet light, because we put the green fluorescent protein gene in there. So that's for the what I call the cool factor. So next week when you look at your bacteria, I want you to promise me to do this. When you look at it, I want you to say, wow, look at that, cool. Because that's what I want to get out of you, right? But in real life, the GFP gene, what does that do? Nothing. I would replace the GFP gene with something more useful, like the human insulin gene, for example. I would instead transform a gene that is more useful for us, like the human insulin gene, so that I can produce it in mass quantities to inject it into our patients. And that's how human insulin gene, human growth factor, and all that is produced this way. But in the lab, I want to give you that cool factor there. So we use the GFP gene, so you can actually see it. Okay, here's another one. There's another way of bacteria acquiring new genes. This one is amazing. The scientist who discovered these won a beautiful $1 million Nobel Prize. So she figured it out, she found them, now she's happy living somewhere with a $1 million. Do you think they stop research? No, they don't. These people that win these prizes, they keep working, of course. I just want to win one. Anyways, I keep thinking and thinking, nothing comes out. All right, so anyways. But this is amazing how, these guys are actually called jumping genes. And they live up to their name. They go jumping around. So this gene, for example, decides, I'm so sick of being here, I want to go there. So it jumps there. One time you find it here, Another time you find it here. So it moved from here to there. That's what it is. That's why it's called jumping genes. Sometimes it says, you know what? I'm so good, I want to make copies of myself. So they reproduce. It reproduces. You don't have just one now. You have 4 or 5 of them. So if this has an antibiotic resistant gene, now you have bacteria that has 4 of them. If this is a little bit resistant, this is for sure resistant. You see that? Sometimes it says, you know what? I want to just go somewhere else. Completely. I'm leaving. That would be this one. So those are called jumping genes. They're also called transposons. Now, for transposons, you have a repeat element on this side and another repeat element on this side. So watch over here, please. This is, for example, the chromosome. When you look and find the transposon in the chromosome, you're going to realize something. There's a repeat element and a repeat element, and the segment in between them is the transposon. This is the rest of your chromosome. This is the rest of your chromosome. So this region here would be referred to as a transposon. The smallest one is the insertion element, i.e., all it's made up of is a repeat element, and a gene called transposase. It makes an enzyme that takes this out of here and moves it there. You see what I mean? That's the jumping gene. So watch. Here's the insertion element. There's actually a gene inside there that makes an enzyme. Then the enzyme will come, okay, we're moving over here. Or we're going to another chromosome or somewhere else. That's what's called the jumping gene transposon. So the shortest ones, the smallest ones, are the ones that are made up of simply this. The repeat element, and the gene that makes the enzyme that recognizes the 2 repeat elements and removes this part and moves it somewhere else. But then there are more complicated ones. Oh, there's so many different kinds. There are more complicated ones that have this, the antibiotic transposase gene, but for example, the antibiotic resistance gene. So some transposons have the minimum parts, but then they have other genes in there. For example, antibiotic resistance. So when this guy jumps, it's taking the antibiotic resistant gene with it. So whether it duplicates or it leaves altogether. So this one here, for example, this is antibiotic resistant because it has a transposon with an antibiotic resistant gene. But now it's no longer antibiotic resistant because this guy left. But then wherever it went, that one is now antibiotic resistant. Or this one here is a little bit antibiotic resistant, but it copied, so now this one became more antibiotic resistant. So anything can happen. Did you know that about 40 to 50% of your DNA are these guys? Think about that one. They're old viral infections, because these are old viral infections, and I'm not talking about a virus that you had last year. I'm talking about a virus that our species had over the years of evolution. We've accumulated all these old remnants of viral infections. And there's a theory saying that the reason why we're here is because of that. They played a major role in our evolution. Think about it. 50% of your DNA are jumping genes. Fortunately, they stopped jumping. Thank God. Most of our jumping genes lost the ability to jump. And I said most of them. I didn't say all of them. But most of them lost the ability to jump. We also have something called retrotransposons. These are old retroviral infections. Remember the retrovirus? It comes in as what? Like HIV is a retrovirus. It comes in as what? RNA or DNA?
DNA.
It actually comes in as RNA. then becomes what?
DNA.
Then shoots for your nucleus, right?
Yeah.
For integration. And then it can sit there and sit there and sit there, and over time it loses its ability to make HIV and all that. I mean, it's an old virus, but it still has the ability to transcribe and then go back to DNA. So these guys here are old retroviruses that are found in our genome. They have the ability to copy themselves. In your genome, they're DNA. But then they're going to do this. They're going to make RNA and then back to DNA, and that new DNA will go and find another home. So it will transcribe into RNA, reverse transcribe into DNA, and that new piece will now go find another home. Now, I don't know where it's going to end up. Depending on where it's going to end up, that will determine the effect of that. Imagine if it ends up in a promoter region controlling your cell cycle division, oh, you're in trouble. Because it's random. It can cause a lot of damage for you, of course. So, here are some of the consequences of these transposons. General effect of transposons. The simple thing is, it scrambles your DNA. It creates new varieties. Again, the theory is that they were involved in our evolution, and actually evolution of all organisms. So, scrambled genetic language. That's as simple as, it's similar to taking the deck of cards and shuffling it. You never know what you're going to end up with. So some of the scrambling can be either beneficial or harmful. It all depends on where the transposon is going to end up. If it ends up in a region that does nothing, then you'll be fine. If it ends up in a very critical region, a control mechanism where, oh, then you're in trouble. It also depends on what is being moved, what is moving, and what does it contain. Because some of these transposons have genes in there. For bacterial, the effect on bacteria for these transposons, they can literally change the morphology of the colony. They can change the pigmentation of the bacteria. Most importantly for us, they can change the drug resistance. Again, this bacteria was not drug resistant until the transposon jumped in there and made it drug resistant, because the transposon came with the drug resistant gene. So anything can happen here. This one escapes a lot of students. Pay close attention, please. Obviously, it's on the exam. There's an inadvertent effect on the DNA. When the transposon leaves, your cellular machinery is not going to sit there, oh, it's okay, we're going to have a gap in the chromosome, it's all right, ignore that. No, it's going to fill it. It's going to say, when the transposon leaves, it says, we can't have that space there. We have to fix that part. So watch up here, please. Look up here, please. Let's say, by random chance, there's a genetic mutation right here. This is a genetic mutation, has nothing to do with the transposon here. But the genetic mutation is right here. The transposon is right here. The transposon leaves. Your cell says, I can't leave this area blank. I'm going to have to fix that. So, let me chew this back and now fix it. So, as it's fixing the gap of the transposon, it has to chew the DNA back, including that mutation. And then when it goes and fixes it, it also fixes the mutation. Oh, that's a good effect. It's not intentional. It's not targeting the mutation. It just happens that the mutation is right next to where the area where it has to fill the gap. And it has to chew back before it starts copying, you see? So it's going to chew back the mutation and fix it. That's pretty cool. Now, what if the mutation is all the way over here? It's not going to do that. It's not going to chew all the way back there. It just chews back a little bit, and if the mutation happens to be there, it will fix it while it's fixing the gap. But listen, it could also introduce a new mutation. Do you agree? It's an enzyme. It can make mistakes. It's not intentionally fixing the mutation. It's as a consequence of filling the gap, and the fact that it has to chew back to fill the gap, it catches the mutation and fixes it. Good. But while it's making the new piece, it can introduce another mutation by mistake. So anything can happen when these things happen. And it's part of that scramble, new genetic mutations, new genetic varieties, and so on.